Welcome aboard Rocket Ship, the platform with the most relevant content for competitive examinations. This is Rocket Ship IAS Lab and continuing with our series on answer writing for general studies papers of civil services main examination. We take up a question that you might come across in GS paper 3. Here goes the question. The 2023 Nobel Prize in Physiology or Medicine was jointly awarded to Katalin Kariko and Drew Weissman for their work leading to development of effective mRNA vaccines. How have mRNA vaccines aided the fight against viral diseases? Briefly explain the mechanism of working of mRNA vaccines. So you can understand this, that the question is quite a surprising one, that this is actually a surprise that sometimes UPSC throws at you. But there have been actually some instances where UPSC has indeed talked about current scientific developments. And an example from you could say quite recent times is the 2021 paper, which asked about the development of light emitting diode and how it had impacted human life. Actually, the inventors of the light emitting diode, Suzuki, Amano and Nakamura, were awarded the 2014 Nobel Prize in Physics. So these questions, that is some current developments in science, are always expected. And what makes the 2023 Nobel Prize for mRNA vaccines more relevant for our current times is that these vaccines were used against the coronavirus. The vaccines were used in fighting the coronavirus pandemic. That makes this type of question, this particular question, quite relevant to, you could say, current affairs. Anyway, we are introducing our answer with briefly defining messenger RNA or messenger mRNA. mRNA. Messenger ribonucleic acid or mRNA is a single-stranded RNA molecule that carries genetic information from DNA to the ribosomes for protein synthesis. Please recall that ribosomes are site for protein synthesis. Ribosomes are organelles, actually cell organelles, responsible for protein synthesis. And what mRNA does, it carries genetic information from DNA to the ribosome for protein synthesis. mRNA vaccines involve, now we would briefly define their mechanism, that is the mRNA vaccines involve first identifying sites or sequences on mRNA coding for specific proteins. These strands or sequences are encased in suitable carriers or vectors. These carriers are sometimes called vectors as well. And this vector, this carrier, is then administered to a healthy person who is being vaccinated, and antibodies bind to the specific antigen proteins, giving rise to immune response. As the carrier already has this sequence for this particular protein is manifested, and this is manifested as an antigen, and the body's antibodies, the healthy body's antibodies or immune cells, bind to the specific antigen protein that is manifested by the carriers, and it gives rise to immune response. Now, even when the immune response has subsided, that is, it has been complete, some lymphocytes or immune cells called memory T and memory B cells acquire the capability of identifying a future infection and bind to the specific antigen to lyse or break down the pathogen. The very name of these particular immune cells makes out their purpose. That is, they are called memory T and memory cells. They keep a memory of the particular infection, and what happens if the same pathogen infests the body in future, these memory cells can actually mount a quick response because they can actually identify this particular pathogen, actually its antigen, its surface antigen. This acquired immunity helps healthy body mount a quick response against a future infection and basically treat it, basically suppress it rather. Now, one more thing we would like to add here, that mRNA is considered safer than vaccines which use live attenuated pathogens. Please recall that some vaccines use live attenuated, that is weakened pathogens. For an example, the oral polio vaccine, it used the live attenuated pathogen, as there is no possibility of the pathogen, that is the weakened pathogen, reverting to its virulent form and causing serious infection. The mRNA vaccine does not contain the pathogen per se. It rather contains a, you could say, mRNA sequence from the pathogen. So there is no fear, there is no chance that this mRNA vaccine would actually cause even a mild disease. Now, graphically representing the mechanism of mRNA vaccine, what actually happens, consider this is the virus and these are the spike proteins. What we first do, I first identify the sequences or codes for the spike protein. This specific protein, we identify the sequence. Then we replicate basically the sequence, or rather separate this, and this mRNA strand for spike protein is then administered or packaged, it is packaged into lipid nanoparticles because they are quite inert themselves. The lipid nanoparticles, they cannot cause infection because they are inert actually. And this carrier, this lipid nanoparticle packaged with the particular mRNA sequence, is then administered as vaccine to healthy cells. And as the carrier, as the vector already has sequences for these spike proteins, they are manifested on the infected cell. The healthy vaccinated cell, it manifests antigen spike proteins. And what happens, the body's immune cells, they bind to these spike proteins. You can understand here very well that this is the mechanism of binding. This is called antigen. This is generally called antigen antibody complex. This binding, you could say, site, and immune cells, they bind to the spike proteins and lyse or break down the infested cell. This way they actually cure, to some extent we can say that they cure this particular infection. But what happens, even after the immune response has subsided, some memory B and T cells still remain, and this is called acquired immunity. This is the entire mechanism of vaccination, but here it is specifically dealing to the mRNA vaccines. And these memory B and T cells, they are prepared for future infection. Whenever the same pathogen for which we have been vaccinated infests our body in the future, these memory B and T cells, they can mount a quick response, and that is mostly successful in, you could say, preventing the disease. That is all mRNA vaccine is all about. Now coming to the efficacy of mRNA vaccines against various viral diseases. mRNA vaccines have been deployed against various viral diseases like Zika virus, chikungunya virus, mini influenza, etc. One of the examples from, you could say, more recent times is the Pfizer's vaccines against SARS coronavirus 2, which was responsible for the coronavirus pandemic. Actually, some of the Pfizer's vaccines were even against the Omicron variant. So there were multiple Pfizer vaccines. It was developed through messenger RNA based mechanism. Basically, this was an mRNA vaccine. Moreover, in the case of India, an indigenous development was the Department of Biotechnology aided COVID vaccine, which was an indigenous vaccine developed by Pune based Genova. What is of note about the Genova's vaccine, that the Genova's vaccine remains stable in fluctuating temperature, which is quite a problem in India, and this reduces the need of refrigeration. Basically, the Genova's vaccine was found to be efficacious even in India's, you could say, heightened temperatures. Now, not only the mRNA vaccines have been found to be effective against viral diseases, there have been some candidates, some experimental candidates, mRNA based immunotherapy, which has been found to be effective against some cancers like prostate cancer as well. So basically, while the question asks you about the efficacy of mRNA vaccines against viral diseases, we can actually go on to add that there have been some, you could say, promising start with using the mRNA vaccine against various cancers. So basically, the mRNA vaccines have the potential of revolutionizing not only antiviral medication but also cancer treatment or oncology. This would obviously save countless lives. Anyway, before we end, a quick recap of the mechanism of the mRNA vaccines. It starts with first identifying of the mRNA sequence in the pathogen, generally the virus. Then what happens? We replicate this mRNA and we introduce it to some suitable carrier or vector, and this suitable carrier or vector is then administered as vaccine. And as the vaccine, as the suitable carrier and vaccine, it already contains sequence for the particular antigen. The antigen is manifested, and this means the body's immune cells can actually mount a quick immune response. And even after this immune response has subsided, it has ended, some of the acquired immunity remains in the form of memory T and B cells, which actually provide immunity for quite a long time, running into even decades actually. So this was all on the mechanism of mRNA vaccines and how they have been successful against many viral diseases and how they have the potential of revolutionizing cancer treatment as well, as some, you could say, successful candidates, potentially successful candidates, have been found to be effective against some cancers as well. This was all on mRNA vaccines, their mechanism and their efficacy. This is all for today. We would reassemble for the next lecture in a few days. As usual, we wish you all the best. Thank you.